Hi everybody, we're picking back up with organic compounds. This is part 2 of the PowerPoint slideshow. We're going to be picking up with proteins, and that is in your course pack, page 22. So proteins have building blocks or monomers known as amino acids. There's about 20 amino acids known to us on Earth, and their combinations create a variety of proteins from hair to fingernails to the keratin or covering that's in your skin. The function of proteins is varied. Some proteins provide structure, others are functional. They exist as hormones or enzymes. And so you can see in the list below, some of these may be familiar to you. Muscle, of course, is a well-known protein, as are hair and nails. But we also know that we're going to talk about plasma membrane structure, how hormones behave in Bio-211, and how enzymes work as well. So this is not a comprehensive list, but this is just a beginning of a look at what proteins can do in the body. Now, proteins are joined by peptide bonds, and these peptide bonds you can see here have carbon, hydrogen, oxygen, and nitrogen. These peptide bonds hold amino acids together. That's why when your body breaks down proteins, which it does every single day, this nitrogen compound is released into the urine. So the ammonium that we smell from, say, urine has its roots here in these peptide bonds holding together these proteins. You're looking at a protein structure here. This protein has many amino acids joined together. They call this a beads on a string look, and that's what is classic of primary protein structure. So you'll notice lots and lots of amino acids joined together here to create a simple structure. Now, proteins often don't stay in this structure. They're going to fold maybe 2, 3, 4 more times in order to receive a final structure. Now in this picture, you can see the beads on a string appearance up here in primary structure. I'm going to represent that by 1 with a degree symbol. This is a polypeptide because it does contain more than 2 amino acids. Each one of these beads is an individual amino acid. The secondary structure you can see here is actually creating a beta pleated sheet that you can see here on the left, or an alpha helix, which looks like a curl on the right. So you can see this particular protein is going to adapt a beta pleated sheet for one region and an alpha helix for yet another region. We then see tertiary structure, which means we have a more complex degree of structure, and there's more twisting that goes on here. And to you and me, this may look like a pile of ribbon with not a lot of meaning, but to the protein, this very much is important because it drives the function of this protein. Rather, the function is driven this structure. So the tertiary structure may lead to a quaternary structure, which is multiple polypeptides coming together. In this image, they have color-coded them: yellow, purple, orange, and green. And so that is the most complex form of protein structure if you get to that level of having a quaternary level protein. Here are some quaternary level proteins. On the left, you've got collagen, which forms the basis for some connective tissues in our skin, for instance, and hemoglobin, which carries oxygen in the bloodstream on the right. So both of these have multiple polypeptide strands that form the protein structure. So you probably are familiar with some of these food examples of proteins, everything from meats and fish to dry beans and nuts. If you're a strict vegetarian, a diet of rice and beans can help give you those 20 amino acids that you need per day. In fact, 8 of those happen to be essential amino acids. Let's turn to page 23 and talk about metabolism and enzymes. We know that enzymes are a special type of protein. They are a particular type that helps to catalyze or speed up chemical reactions. So chemical reactions in the body form the basis for metabolism, which is known as the sum of all chemical reactions. We've already talked about catabolic reactions that break down polymers into monomers, and we've also talked about anabolic reactions, which build monomers into polymers, and enzymes can assist with both of those types of reactions. So what you're looking at here is just a generic enzyme. You'll notice it has an active site that looks to be a pretty special fit with that substrate. We call this a lock and key fit because the fit goes together so well. In fact, this enzyme won't work on any other substrate except for the one that it's built to work on, like you see here. Enzymes can do a little twist to make sure that they fit with their substrates, but they, for the most part, don't work on any other substrate except for the ones that they're built to work on. Now, if this substrate was a sugar, you would know it has an ending of OSE, like maltose has an ending of OSE, and this enzyme, being the one that works on maltose, would actually have an ending of ASE. So ASE as a suffix tells me that's a particular enzyme that works on this prefix. So that kind of gives you a powerful tool. Now we know that cellulase is the enzyme that we lack to work on sugars known as cellulose. Let's talk for a moment about activation energy. This is the amount of energy that is required to make a chemical reaction run. We talked about this earlier in Chapter 2 when we discussed chemical reactions. My analogy then is the same here. Imagine that you have a 400-pound lead ball and it is your job to push it up and over this hill. By yourself, you would have the without enzyme hill. That's a lot of work for your body to accomplish that chemical reaction. That means that much energy has to be put into the reaction in order to make it run. However, with the help of an enzyme, the enzyme chops the hill down and so makes your job easier. So with the enzyme, you don't have to work quite as hard and you don't use as much energy to get the chemical reaction to start. So you can see here that enzymes assist with the activation energy for a chemical reaction. Let's take a look at this chemical reaction. In this reaction, we can see the active site here, which is the special point on the enzyme that fits with the substrate here. You can see when they fit together, we can perform whatever chemical reaction is needed. And here we're going to break apart that reactant into two products. So thinking back, what type of chemical reaction do you think is shown here? Okay, so this type of reaction is a breakdown reaction known as a hydrolysis reaction. I should also mention here that if we change the shape of this enzyme through a process called denaturation, that this enzyme will no longer meet with its substrate. The active site will be changed, and unfortunately that means that there is no longer a chemical reaction that can occur. You'll notice in your worksheet that there are several factors that can influence enzyme activity, including an acidic pH, heat, and concentration of reactants and products. Now, let's focus on heat and acid for just a moment. If you're female and you do chemical treatments to your hair, you may use acids to provide curls that were not there, or you may use heat to straighten your hair or to curl your hair. So that's an example of how we can use heat and acids to change the shape of proteins, although with hair it's a temporary change, not a permanent change. Alright, so I'll give you a moment to take a look here and see if you can identify the enzyme, products, active site, and substrate. Alright, so we know that ASE is an indicator for an enzyme. So lactase here is an enzyme. The substrate it's acting upon ends in OSE. That happens to be a sugar. So this particular substrate happens to be a sugar. That may not always be the case. Where the two join together is called an active site. And what we create over here are the products. And this again happens to be a breakdown reaction or a hydrolysis reaction. So let's turn our attention to the last organic compound group called the nucleic acids. This particular group has a building block that contains 3 parts. Each nucleotide that forms a nucleic acid has a phosphate group, some type of sugar, and a nitrogenous base. So we'll dive more into details of those as we cover each example. The function of the nucleic acids is to store genetic information as DNA or RNA and to serve as an energy molecule as ATP. And before we go any further, you may wonder why in the world we've got such a variety of examples here, and it's because they're all built from the same basic nucleotide, with either phosphate, sugars, and nitrogenous bases forming the basis of that nucleotide. take a look at DNA. DNA is deoxyribonucleic acid, and it is our genetic blueprint found in almost all of our cells. Let's talk about a nucleotide of DNA, first by looking at this structure here. The phosphate group is represented by a P. This is a 5-carbon sugar, and I know it's 5-carbon because it's got 5 sides to that ring. This particular sugar is called deoxyribose. Hey, there's that OSE ending that reminds us it's a sugar, and this is the D in DNA. There's also a nitrogen-containing base, and here it's represented by G. So there are 4 different bases here. You'll notice A's, T's, C's, and G's. Now, the sugars and phosphates form the backbone of the DNA molecule. You can see they repeat phosphates and sugars over and over up each one of these backbones. The rungs of the ladder, you might say, are formed by these nitrogenous bases, adenines, thymines, cytosines, and guanines. You may also recognize these dotted lines in between each of these base pairs. Those are hydrogen bonds. You may recall we talked about those previously in this chapter, and they are represented by dotted lines when we see them. So let's just circle another nucleotide. Right here's a nucleotide that contains adenine. Because each nucleotide contains a sugar and a phosphate, the only thing that makes one nucleotide different from the other is the base. So when we talk about nucleotides of DNA, we refer to them by their base because that's what makes each one different. So when I talk about thymine, I'm talking about this nucleotide because it contains that particular thymine as the nitrogenous base. By contrast, RNA is only a single-stranded structure, and it has a few differences. For one, you'll notice that we have a different sugar here known as ribose. We still have a 5-carbon sugar, it's just ribose this time. We still have the phosphate group, okay, but the bases here are slightly different. Instead of having A, G, C, and T, uracil replaces thymine. Okay, so keep in mind that uracil, or U, is only found in RNA molecules, while T, or thymine, is only found in DNA molecules. So DNA is a copy of RNA, and it is used to help us build proteins. This particular molecule can leave the nucleus of the cell where DNA is stored and travel into the cytoplasm. You know, if you kill the photocopy, it's not a huge deal because you still have the DNA in the nucleus that you can always go back to and make another copy from. So we think of RNA as a copy of our DNA molecule. Here, side by side, you can see a comparison of DNA on the left, which is called a double helix, and RNA on the right, which is a single helix. Some people describe the DNA molecule as looking like a spiral staircase, and it really does. What makes up the backbone? The backbone is made up of sugars and phosphates. And what makes up the rungs of these ladders, like you see here? Those are the nitrogenous base pairs. Adenines pair with thymines, cytosines pair with guanines. Finally, in this category, we have adenosine triphosphate, or ATP for short, and that stands for adenine, the base here. That is the only base we have in this molecule. Ribose, the sugar, similar to RNA, right? But this time we have 3 phosphate groups. That's where the tri comes from, and ATP, the triphosphate. This is our energy molecule. We'll explore a little more about how this works in a later portion of Chapter 3. But this particular molecule is our energy currency in the body. Think of it like a debit card for providing energy whenever your body needs to do work. So you may think again, why is this particular molecule with a different purpose in this category? Well, ATP is still made up of a nucleotide. Nucleotides have 3 components. What are they? A sugar, happens to be ribose here, a base, which happens to be adenine here, and phosphate groups, which we do happen to have 3 here. So just a moment for you to take a look at these questions to review our organic compounds. And then I'll provide the answers after you've had a moment to think about these. And I'm going to encourage you to pause your video for just a moment so that you can write down all these answers. All right, so the building block of carbs is a monomer called a monosaccharide. Glucose is a great example of a monomer. The three types of nucleic acids are DNA, RNA, and ATP. Glucose is stored as glycogen in the body in two places. Where are they? Liver and muscles. There's many differences between saturated and unsaturated fats, but one major difference is the bonding. Saturated fats have only single bonds, while unsaturated fats have one or more double bonds. Another example may be the fact that saturated fats are solid at room temperature, while unsaturated fats are liquid at room temperature. And finally, most saturated fats are produced by animals, while unsaturated fats are produced by plants. Polysaccharides, for number 5, could be cellulose, could be glycogen, one more, could be starch. And then finally, number 6, enzymes are classified as proteins. All right, so I hope this helps. I encourage you to use flashcards where you have material that won't stick, and come to me with any questions you've got.